Hello students, I am here to teach you the next lesson in class 10th bio in the chapter life processes. Yesterday I taught photosynthesis, but one more event is there which is left, that is transpiration. And after transpiration, I will teach you nutrition in amoeba also. Today, I will try to cover this heading also. So, you can see here, transpiration. What do you mean by transpiration? As you know, the plant roots absorb water from the soil and the water goes upward and the excess water gets lost in the form of vapor, and this is called transpiration. So transpiration is the loss of water in the form of vapor from the aerial parts of the plant, that is stem, root and leaves. The transpiration. Now the next heading is types of transpiration. Transpiration is of three types. The first one is the cuticular transpiration. The next is lenticular transpiration, and the third one is the stomatal transpiration. If transpiration takes place through the cuticle, it takes place by cuticle. If transpiration takes place through the cuticle, it is known as cuticular transpiration. But the question is, what is cuticle? The cuticle is a thick layer present just above to the leaf, and it has been observed that 5 to 10% of water gets lost through the cuticle also. Now the second one is the lenticular transpiration. Lenticular transpiration takes place through the lenticels. Now there is a question that what are lenticels? The lenticels are the minute, it means the very small apertures present on the bark of the stem, in between the bark of the stem, in the old stem, and less than 1%, not even 1%, less than 1% of water is also gets lost through the lenticels. Now the next one is the stomatal transpiration. Stomatal transpiration, most of the water get lost through the stomata, and this is called stomatal transpiration. And stomatal transpiration is included in class 10th syllabus. These two, transpiration, cuticular and lenticular, have been not added. So we will discuss especially about the stomatal transpiration, structure of stomata, and how stomata opens and gets closed. So let us start the stomatal transpiration in detail. Stomatal transpiration. Structure of stomata. Now, what are stomata? Stomata are the minute apertures, or you can say the minute openings present on the surface of the leaf. The stomata you can observe only under the microscope. In dicot leaf, stomata are present on the lower epidermis, but in monocot leaf, the stomata are present on both of the surfaces, on upper and lower epidermis. Stomata are kidney shaped in dicot leaf, in dicot plant, while in monocot plant it is dumbbell shaped. Each stomata has the following components, following parts, and these are guard cells, stomatal pore, and the third one is the subsidiary cells. You can see here the diagram of the stomata. This is the diagram of the stomata, which is open. These two are the guard cells, and this aperture is stomatal pore just around the guard cells. Some of the mesophyll cells are there, and these neighboring, you can say, the mesophyll cells which are near to the guard cells, these cells are called subsidiary cells. The stomata has the two walls. This is the outer wall, and this one is the inner wall. The inner wall is very thick. This wall is very thick, and the outer wall is thin. Outer wall is very thin and inner wall is very thick. The thickness and this thinning of the outer wall helps in the stomatal movement. I will cover in the next study. So overall, these all are the components of stomata. That each stomata is made up of two kidney-shaped guard cells and has a stomatal aperture. This is called stomatal pore. This is also known as stoma. And the mesophyll cells of the leaf which surrounds these two guard cells, these mesophyll cells are called subsidiary cells. And in this diagram, I have shown the opening condition of the stomata. Now, I will start the opening and closing of the stomata. Opening and closing of stomata. As you know, stomata opens during the daytime and it's get closed during the night time. Now there is a question that why stomata opens during the daytime and gets closed during the night time. During the daytime, in the presence of sunlight, some changes takes place inside the guard cells. The guard cells then open, and this opening of the guard cell and closing I can explain here by a theory that is called starch sugar conversion. Starch sugar conversion. You can see here, suppose this one is the stomata and this stomata is closed. This one is the closed stomata. Now I will explain that how this stomata in the presence of sunlight during the daytime it opens. During the daytime, the starch, it's very very important. Suppose this one is the starch. The starch is converted into the sugar, and you know starch is insoluble in water and sugar is soluble in water. When the starch is converted into the sugar, the osmotic pressure of the guard cell increases. It means the guard cells, it become what? It becomes hypertonic. Hypertonic means more solute and less solvent. Now the water moves from neighboring cells, from the subsidiary cells, from the outside to the guard cells. The guard cells become turgid, and when it becomes turgid, a pressure is created on the wall, on the outer wall, and this wall is stretched and stomata opens. So during the daytime, starch is converted into the sugar. The osmotic pressure of the guard cells increase and osmosis takes place. The water moves from outside to the inside. The guard cells become turgid and stomatal pore opens. During the night time, what happens? When there is no light, then the sugar is again converted into the starch. The osmotic pressure of the guard cells decreases. It means the guard cells become hypotonic. It loses water. Water moves where? Outside. And the guard cells become flaccid and stomatal pores gets closed. So overall, it is not very detailed in class 10, but you have to remember, you have to learn that the opening and closing of stomata depends upon the turgor pressure of the guard cells. When the turgor pressure of the guard cells become high, stomatal pores, stomata opens And when the turgor pressure decreases, the osmotic pressure decreases, the stomatal pores get closed. And this is overall the opening and closing of stomata. Now, functions of the stomata. It's very, very important. Functions of stomata. What are the functions of the stomata? The first one, it helps in the process of transpiration. As I told you, transpiration is the loss of water in the form of vapor from the aerial part of the plant, and especially through the leaves, the excess water gets lost outside through the stomatal opening. So it removes excess water from the leaf, from the plant, and it helps in the process of transpiration. The second, it helps in gaseous exchange during the process of photosynthesis. It takes carbon dioxide, and the carbon dioxide is assimilated to form glucose. So it helps in the process of gases exchange also. So these two functions are there. That is, it helps in the process of transpiration, as well as it also helps in the process of gases exchange. If anyone is asking that what are the importance of transpiration, so you can say that transpiration removes excess water from the plant, and transpiration also brings cooling effect to the plant. When the water gets lost, it brings cooling effect. And one more function of the transpiration, when the water moves upward through the xylem, it also helps to translocate, or you can say for the movement of water and minerals from downwards to the upward. I will explain later on transpiration pull theory. In this heading, I will teach you in detail that how transpiration helps in the process of upward movement of water and how minerals goes upward. So this was overall about the transpiration. Now one more heading I will explain today: nutrition in animals and especially in amoeba. Nutrition in amoeba. There are two headings: nutrition in amoeba, two topics are there, and nutrition in human beings. Both the types of nutrition, that is amoebic nutrition and the nutrition in human being, are included in class 10th. So let me just start the first one, nutrition in amoeba. And yesterday I taught about the nutrition, and I explained the definition of nutrition, that nutrition is the process of the intake of the food, its digestion, absorption, assimilation, as well as egestion. And especially amoeba and human beings and other animals also, we all perform what kind of nutrition? Holozoic mode of nutrition. Amoeba performs holozoic nutrition. In holozoic mode of nutrition, as you know, the animals, including the human being and amoeba, take the solid food from outside. This is called ingestion. Then digestion takes place. Then digested food gets absorbed. Then they are get assimilated, and then undigested food materials are removed, and that is called digestion. So all these headings you have to make the point: ingestion, digestion, absorption, assimilation, and the last one is the digestion. The holozoic mode of nutrition in amoeba you can understand with the help of diagram. This one is the amoebic cell. And you know, amoeba is a unicellular organism, protozoa, present in the water, and it takes the solid food from outside. Some of the phytoplanktons, zooplanktons are their food. This one is the food, and this is the amoeba. This is cell cytoplasm. This one is the nucleus. This is the structure of amoeba. When amoeba is in the contact of the food, to intake the food, it forms a structure that is called pseudopodia. What is the name of this projection? This is called pseudopodia. Amoeba don't have the mouth. It takes the food with the help of these two finger-like projections, and this is called pseudopodia. So pseudopodia, or the invagination, you can say, the finger-like projections which helps in the process of ingestion or to intake the food. And pseudopodia is also a locomotory organ. It helps in the movement also. The pseudopodia trap the food. Then it goes inside this vacuole. Pseudopodia actually inside towards the cell cytoplasm. It forms a vacuole-like structure. This is called vacuole. Later on, the pseudopodia disappeared, and this vacuole comes inside along with the food. This is the food vacuole. The process of ingestion is completed here. Now, after the process of ingestion, the process of digestion takes place. This is the food vacuole, and this complex food is broken down into the simpler food, and this is called digestion. How the process of digestion takes place, from where the enzyme comes, let me to explain. When the food vacuole comes inside the amoebic cell, it fuses with a structure that is called lysosome. And lysosome is filled with the hydrolytic enzymes. All the digestive enzymes, or you can say the hydrolytic enzymes, are contributed by the lysosomes, and these enzymes are required to convert the complex food into the simpler food. And this is the process of digestion. After digestion, the digested food goes outside, that is from food vacuole to the cell cytoplasm, and this process is called absorption. This is called absorption. And after absorption, as you know, the food materials are utilized by the body cells. Here by the amoebic cell, it is a single cell in a structure. The cell for the different functions, for getting the energy. The food is also required for the construction of the cells, to repair the cells. This is called assimilation. And the undigested food materials inside the food vacuole, not all the food materials are digested. Some of the food materials, they are undigested, and these undigested food materials are removed outside to the amoeba, and this process is called egestion. During the process of egestion, the amoebic cell don't have any special point. At any point of the amoebic cell, the cell membrane gets ruptured, and the food vacuole along with the undigested food materials are removed outside, and this is called ingestion. So this is all about the holozoic mode of nutrition in amoeba. Once again, you can see in these three headings, you can in these three diagrams you can see how amoeba is getting food from outside. This is the solid food. To intake the food, amoeba forms a temporary structure. This is called pseudopodia, and with the help of pseudopodia, it captures the food, takes the food from outside, it forms the food vacuole. After ingestion, the food materials are digested, means broken down into the simpler form, and this is called digestion. After digestion, the undigested food materials are removed outside to the body, and this is called egestion. The digested food materials are utilized for the different functions, and this is called assimilation. So this one, the diagram is very important, and you have to make the diagram, you have to practice. You can see the diagrams are already given in the NCERT book. You can take the help of some reference books also, but the diagram is very important. Now tomorrow I will teach you the next topic, nutrition in human beings. Before I close this lesson, I am giving you some assignments. Please note down. Please note down some of the questions. You note down in your here copy, either any rough copy. What is transpiration? Write its significance. Second, make a level diagram of stomata. Make a level diagram of stomata and explain about all its components. Question number 3. Write the functions of stomata. Write the functions of stomata. Next, name the mode of nutrition which takes place in amoeba. And the last question is, explain the process of nutrition in amoeba with the help of diagrams. Explain the process of nutrition in amoeba with the help of diagrams. Thanks.